talking headphones at the same time. So we started chapter 12, right, talking about inheritance, right? So chapter 11, we covered meiosis, how chromosomes behave, what meiosis is, why it happens, how it works. And then in chapter 12, we're seeing kind of, because of everything that happens in meiosis, right, ends up being why we see kind of everything we see as far as what we inherit and how traits work and how traits get passed on, right? So we started talking about dominant and recessive traits, and we talked about Mendel and his work and how it's kind of revolutionary in the sense that he just wrote down what happened, brilliant, counted things, fantastic, right? And so we ended on these conclusions of Mendel's work, right? He's working with these pea plants in this little abbey in Austria. I'm sure it was gorgeous. Right, and so he came to these 4 conclusions. So we'll kind of revisit these and then we'll start kind of going on through the rest of another chunk of chapter 12. Chapter 12 is kind of a long one. So conclusion 1 from Mendel's pea plant experiments is that the plants didn't produce progeny of intermediate appearance, right? We're talking about like flower color, for example, right? He crossed purple flowers with white flowers and he did not get pink flowers, right? The plants either presented or expressed a gene for purple flowers or a gene for white flowers, right? There's no blending. This kind of traditional idea that traits get blended between parents is not a thing exactly. All right, now he also determined that for every pair of traits, right, that he was looking at, right, green or yellow seeds, purple or white flowers, whatever, one was not expressed in his F1 hybrids, right? So if he crossed purple flowers with white flowers, right, his parent strains, that F1 generation, those first offspring, one of those traits didn't show up, white flowers, right? And so, and we call that the one that showed up, the purple flowers, dominant. And so he determined that because when you bred those together, you got white flowers, that those F1 purple flowers were carrying the trait white flowers, right? Just not showing it, right? So you can have a trait and just not express it, right? Traits are segregated among progeny. That just means that you have some offspring that have one trait, some offspring that have another trait. The traits are kind of allotted among them, right? We didn't have plants that had purple and white flowers on the same plant. The traits kind of segregated among individuals. There we go. 4. When he bred these F1 hybrids together, right, all of these purple flowers, some of which were carrying white alleles, all of which were carrying white alleles, the F2 generation expressed traits, in this case, purple and white flowers, in a ratio of 3/4 dominant, so 3 out of every 4 were purple, and 1/4 recessive, 1 out of every 4 was white, right? And so this 3-to-1 segregation ratio, right, this segregation of traits and this 3-to-1 ratio is known as the Mendelian ratio for monohybrid crosses, right? Because we're talking about monohybrid crosses, right, where we take this one trait with 2 options and we cross 2 hybrids together, monohybrid cross. All right, so his conclusions from the data he collected. Using this data and these conclusions, he created a model of patterns of inheritance, right? So this model has basically kind of 5 tenets, 5 elements to it, and we still use this today, right? It's still applicable, basically. It still works, at least for certain kinds of traits with 2 options and 1 gene, right? So element 1, parents don't transmit traits directly to offspring. Rather, they're just passing the information for the trait, right? Which is not really kind of earth-shattering to us, but if you think about kind of this Victorian style mind where like you have a kid and the kid is just kind of this weird like recreation of yourself that you make, right? It's not really how it works, right? You carry a whole lot of information. You're expressing, right, DNA. He didn't know about DNA, but he was kind of intuiting it even without knowing that it existed, right? You're carrying all these genes, you're expressing some of those genes are just kind of hanging on to, and you pass that information on, and that information creates another human. It's kind of a weird way to think about it, but that's what happens. All right. So every individual receives 1 copy of each gene from each parent, right? So you have conversely, you have 2 copies of every gene, right? And that's that there are 23 human chromosomes. You have 46 chromosomes in every cell because you've got a copy of each from your mom and a copy of each from your dad, right? So you have 2 versions or at least 2 copies of every gene that you've got in each of your cells. All right. And not all these copies are identical, right? Which makes sense. We have variation, so they can't all be identical. We would all look exactly the same, it'd be silly, right? And so there's a term for this. I probably said it by accident, but we're going to actually introduce it officially now. Alternative forms of different genes are called alleles, right? So it's like versions of genes, basically. Versions of genes are called alleles, right? So if you have a flower colored gene, right? In our case, you have in the pea plants a purple allele and a white allele, two versions of that gene, right? Now, more words. Offspring who inherit the same allele from both parents, right? Inherits maybe the purple allele and the purple allele. They have the same copy, right? Same version. And that zygote that's created, they are homozygous for that trait. Okay. Allele from each parent, and they're the same. Now, if you inherit different alleles from each parent, right? So this offspring inherits a purple allele from one parent and a white allele from the other. They've got two different alleles for the same gene. That individual is heterozygous, right? Hetero just means different, right? So their zygote has two different alleles for a trait, right? Cool. So you get a copy of every gene from each parent. Those copies are not necessarily identical. And those two alleles remain independent throughout this process, right? I don't get a purple allele from one parent. I don't know what kind of purple gene I'm getting, but it'd be fun. Purple allele from one parent and a white allele from one parent. And they don't like come together in the nucleus and like zhuzh themselves together. That's not how genes work, right? So the two alleles remain discrete. There is no blending of traits. He was very strong on this point. Tell those Victorians there's no blending of traits. 5. The presence of an allele does not ensure it will be expressed, right? So if a flower inherited heterozygous alleles, right? One purple and one white. Purple is dominant. The flowers will be purple. It's expressing purple. I'm clicky today. The white allele will not be expressed, right? It is recessive. So it's carrying the gene for white flowers, but it's not actually using that DNA. It's just kind of hanging on to it. And it can pass that on, right? We saw that in those monohybrid crosses. So that is Mendels model. For these types of genes, these types of traits, this is true. Cool. All right, let's get some more words. Never gonna have enough words. Let's make it a little bit more. Projector's getting kind of sad. All right, genotypes versus phenotypes. And we have hinted at this as a concept, but we haven't given any words to it, right? Because we talked about you inherit two alleles of every given gene, and you will express one of those. One of those you'll kind of hang on to and not express, which means that the genes you're carrying may not match your physical appearance in its entirety, right? Because you're carrying genes you're not using. So all the genes you have, which is two copies of everything, is your genotype. It is the type of genes that you have. That's easy, right? So the total set of alleles, right? 2 per for all of them is your genotype. Your appearance, your physiology, the things you're expressing, the genes that you're using, right? Your actual physical appearance, which is the result of what gets expressed of each of those allele pairs, is your phenotype. Right, so phenotype is just the way, basically the way you look. The genotype is all the actual DNA you have, whether or not you're using it. Okay. Right, and so we saw this here, right? We bred purple and white flowers to create this F1. These are all hybrids, right? So they're each carrying a purple allele from this parent and a white allele from this parent. So their genotype, they have an allele of each, right? They're heterozygous. They inherited a white allele from this parent, they're just not expressing it, right? So their phenotype is purple, but their genotype actually has a purple and a white allele, right? So your genotype and phenotype are related, but they're not necessarily completely the same. It works for true breeding things, right? This white recessive figure here, right, inherited 2 white alleles. So their genotype is only white alleles and their phenotype is white, right? but it kind of lets us talk with a bit more precision about what something looks like versus what kind of genes it's carrying. All right. So this gets us to Mendels Law of Heredity, or the principle of segregation. So principle of segregation states that the two alleles for a gene, right, you've got two copies of everything, segregate during gamete formation, right? Remember, because during meiosis, we separate our maternal and paternal versions of the chromosomes, which means we have these two alleles, these two versions, and they're not going to go anywhere together, right? In meiosis, we take them apart and put them in different cells, right? So two alleles for a gene segregate during gamete formation, i.e. meiosis, and then get rejoined at random, right? A gamete from this parent and a gamete from this parent, which has one copy of every option, right? Fused during fertilization, right? So you get this principle of kind of this segregation of traits during gamete formation. This is based on the behavior of those chromosomes during meiosis, specifically metaphase 1, right? That independent assortment is what causes this to happen, right? Where we split maternal and paternal homologs apart into separate gametes or haploid cells. You've got this. All right. We knew they were coming. Moving on. Punnett squares, right? You knew they were going to happen. They're not so bad. Well, they're going to make sense. We're going to make them make sense, right? So they're going to make sense. I believe in you. Punnett squares allow us to symbolically analyze, it's like freaking warm in here, patterns of heredity. Yeah, inheritance, right? So what we're going to do, remember we've got two alleles. Every individual has two alleles for every gene. In the case of the traits that we're looking at, we have options to have one of them be dominant and one of them be recessive, unless we have two of the same, right? Dominant traits, right? If you have the allele for purple flowers, right, it'll be an uppercase, a big letter, capital letter. If you're carrying the recessive trait, we're going to represent that allele with a lowercase letter, okay? You have two copies of each gene that you get from your parents, and so your genotype for any given gene will be represented by two letters, right? You've got one maternal and one paternal version. So your genotype will be represented by two letters for each trait that we're looking at. Example. So say we're looking at some trait represented P. It's probably going to be purple flowers because that's just what we got, right? So the dominant trait is capital P. The recessive trait is lowercase p. And so this is that F1 cross, right? These guys were purple flowers that were carrying the recessive allele, right? So I'm going to cross one of those that has a dominant and a recessive version with another one that has a dominant and a recessive allele, right? So for every gamete that this individual reproduces, it's going to pass on either a recessive or a dominant version, right?
How do you know which is going to happen?
It's random. But we know what options there are, right? So what could happen? So the mom could pass on a recessive trait. Cool. And maybe the dad passes on a recessive trait. Option 1, we're homozygous recessive. Or the mom passes on a dominant trait. Dad still passes on recessive trait. We're heterozygous, right? Option 2. Option 3, this time, dad passes on the dominant trait, mom passes on recessive trait. We're still heterozygous, just the other way around. And lastly, they could both pass on the dominant trait. Yeah, homozygous, dominant. Right. Yeah. So you can think about it like that, right? They're forming gametes. So, right, so when this individual forms gametes, it will either have this allele or this allele, right? So it's either going to be passing on that one or that one. But it's just filling in a table, right? When you fill in any of these kinds of like little tables, you just kind of just pulling the letters through the table, all right? All right, all right, all right. So you're passing on one allele to each offspring from each parent. So flowers. This is that true breeding parent cross, right? We had a true breeding white flower, true breeding purple flower. it turns out, when we were saying true breeding, what were we actually, what did we mean? This individual is a true breeding purple flower. It means that that flower or that plant is, what's its genotype then? If it's true breeding purple. It only has purple genes. Yeah, it's homozygous dominant, right? So we say true breeding in this kind of amorphous way because Mendel didn't know about genes. But right, if it's only making purple offspring, you can infer that it only has the purple alleles, right? That's why all its offspring are purple, because that's all it's got to pass on. Same thing for the white individual, right? All of its offspring, for the purpose of the white individuals, will be white because it's only carrying white alleles. Now, if you cross these, because the trait purple is dominant to the trait white, all your offspring are purple, right? Because no matter which allele goes into this flower's gametes, it will make purple offspring, right? If you get the purple allele, you are purple, end of story. This one will only be passing on lowercase p's, the recessive trait for white flowers. But if there's a dominant allele present, you won't see that the white trait is there, right? So it's carrying the recessive allele, but showing the dominant one because it's dominant, right? So white flowers were recessive and the purple flowers were dominant. So you end up with this F1 generation that's all purple, but are all carrying the recessive allele. Okie dokie. So
the opposite?
Is which
Because
opposite?
you said it was a big P, but then you said it was lower, right? That makes sense. No, not really.
So purple's capital P, white is little p, right? So white flowers are only passing on little p's. Purple flowers are only passing on big p's.
But just because you have a big P, it automatically.
It like overrides everything. Yeah, if you inherit a big P allele, you're purple. End of story. And so, because this is passing on the same thing to all offspring, and this one's passing on the same thing to all offspring, these are all the same, right? Because they each get one of each. We're each going to get a big P and a little p no matter what happens, right? So, if we breed these together, they're heterozygous, right? They have one dominant allele, one recessive allele, one recessive, right? We do this jobber. So, if they both pass on their dominant alleles, you get a homozygous dominant purple flower. If this one passes on its dominant allele and this one passes on its recessive allele, it looks just like its parent, right? But it's still going to be purple, even though it received a recessive allele. Same thing here. If this one passes on its dominant allele, this one passes on its recessive allele, it's still purple, but they're heterozygous. Lastly, it's possible for them to both pass the recessive allele, right? So if the offspring only gets recessive alleles, then it's white. Okay. So that's the cross we already saw. We already explained. It's just now we've tacked the genotypes in. Right? We saw this on Wednesday. We just stuck genotypes in the corners. Same thing. The same thing we were looking at, right? And this is where you get a 3-to-1 phenotypic ratio for a monohybrid cross, right? Using like all the words we got together in a single thing, right? Because the phenotype is purple, and this phenotype is white. 3 purple to 1 white. The genotypic ratio is 1 to 2 to 1. Right? Okay. Let's make it harder. So we're doing monohybrid crosses as Punnett squares. You can also do a Punnett square for a dihybrid cross. Right? So we've been using one trait at a time, right? Flower color, flower color. We can look at 2 traits at a time, all in the same square. It's fine. So let's look at some dihybrid crosses. This will get us to Mendel's principle of independent assortment, which kind of builds on principle of segregation, right? Yeah, let's do that. So we can look at 2 independent traits in the same Punnett square. Okay, here's what that's going to look like. Let's pick some different traits. So we can look at seed color, right? Seed color, color peas in a plant. They can be yellow or green, and they can be round or they can be wrinkled, right? So two different traits that we can kind of evaluate independently, right? And so in the case of these pea plants, the dominant trait is yellow, recessive trait is green. General convention is that the letter is just the first letter of the dominant trait, right? So we have big Y, little y for yellow versus green seeds. And seed shape, round seeds are dominant, wrinkled seeds are recessive, so we get a capital R for round, and a lowercase r for recessive wrinkled allele. Okay, so we've got two traits, independent of each other. Now, we're gonna add some stuff to these little cross diagrams, but it's fine, right? Because, so this individual, this is our true breeding dominant traits, right? Yellow and round individual, which means that its genotype is big R, big R, big Y, big Y. What that means is that from its parents, it inherited two big R alleles, two big Y alleles. Make this guy. This one shows is homozygous for both the traits, the recessive traits. So it's got two little r alleles, two little y alleles. All right, so two traits. It's homozygous for both traits. All right, so here, this guy, it's got two copies of big R, two copies of big Y. All of its gametes, no matter what, will be one big R and one big Y. All right, that's all it's got. That's all it can make. Same thing here. All this thing can pass on are little r's and little y's. End of story, right? No matter what you cross, they're making the same gametes, which means, since you only kind of have one cross option, all of your offspring will look like this, right? They're going to get these alleles and these alleles, right? Two copies of this one from each parent, right? Copy of this one from each parent. So here is its genotype, big R, little r, really scratchy. Big R, little r, big Y, little y. Okay. It's just like the flowers, right? We just added another set of letters, right? Because we had capital P's and lowercase p's. So we got a big P, little p. We just have two of them. Big R, big Y, little r, little y. It's the same. It's the same.
Are these colors just made up?
I mean, they're actual colors of peas they're actual like pea plant traits. Yeah.
I get thrown off, I see like pink or something.
No. Well, I don't know. There's all kinds of weird pea plants and stuff. For our purposes, we're just looking at green and yellow seeds. Yep. So we have a heterozygous F1 hybrid, right? It's heterozygous for both of our traits. Now, just like the flower F1, which were all purple, this dihybrid F1 will be all yellow, all round, right? It's going to show all dominant traits in the F1 generation. That does not change. The genotype, similarly, is heterozygous for both traits, right? Big R, big Y, little y. Yeah. Yeah. Now, here we go. Because of independent assortment during meiosis, right? The behavior of these different chromosomes, right? My R chromosomes don't affect the behavior of my Y chromosomes. Right? Each of them are doing their own thing, right? So because of independent assortment, my F1 individuals are going to produce 4 kinds of gametes, depending on which ones they pass on, right? So this heterozygous individual could make gametes that have a big R and a big Y. It could have a big R and a little y. You have a little r, a big Y, or a little r, a little y, right? Those are the 4 combinations you could get in meiosis, right? In gamete formation, right? So we have these 4 genotype options, right? Of the gametes that this individual could produce. So we can take these genotypes and put them in a Punnett square and see what kind of offspring crossing 2 of these heterozygous individuals together would produce. And it would look like that, which is fine. right? So all we've done, so these are those 4 gamete options, right? You get a big R, little y, little r, big Y, little r, little y, right? And then you just pull them through, right? Big R, big Y, big Y, big R, big Y, little y, right? And on and on and on and on, all the way through the table. Yeah, just kind of fold them in. And so Mendel's dihybrid ratio ends up being 9 to 3 to 3 to 1. The phenotypic ratio. So we can see that, right? We have 1, 2, 3, 4, 5, 6, 7, 8, 9 round yellow. We have 1, 2, 3 round green, 1, 2, 3 wrinkled yellow, and 1 wrinkled green. Cool, 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 cool. Now, if you stare at this a little longer and a little harder, you will notice that if you separate the R and Y traits out, the 2 genes have acted independently and still each individually have Mendel's 3 to 1 ratio. Right, so we've got 16 squares, so 3 to 1 is going to be 12 to 4, right? Yes. So round versus wrinkled, we should have 2, 4, 6, 8, 10, 12 to 4 wrinkled, right? Because this is our recessive, that's our dominant. We do this color, yellow, 4, 7, 8, 9, 10, 11, 12. Dominant yellow, 4 recessive green. Right, so we've shuffled all the genes together, but because they're acting independently, they still show up in a 3 to 1 ratio, even though we've kind of thrown them all into the same table. The same thing is happening. They're not affecting each other. Yeah? Okay. This is Mendel's principle of independent assortment. Right, so in a dihybrid cross, the alleles of each gene act independently. We're looking at them together for whatever reason, but they aren't affecting each other at all. They're still just doing the same thing they would have been doing if we looked at them in a monohybrid cross, right? So the segregation of different allele pairs, whether a seed is round or wrinkled, whether a seed is yellow or green, right, is happening independently of each other. And that is Mendel's principle of independent assortment. Okay.
Oh.
You'll be fine. All right, we'll sit with it for a minute, right? It's a lot of words. All right, and again, just like the principle of segregation, this is tied to the behavior of chromosomes in metaphase 1 of meiosis, right? Everything has to do with the way those chromosomes line up, where we separate maternal and paternal homologs, where we separate alleles out, right? Everything basically about how you inherit traits from your parents has to do with meiosis. Okay. Yeah. Let's see how far we get into this. So, math sidebar, brief, superficial, it's fine. So, we have looked at, right, just the phenotypes of plants, right? Mendel bred a bunch of flowers together. We looked at purple and white flowers. That was great. We looked at Punnett squares, right? We can then put those, kind of layer those crosses over Punnett squares and kind of symbolically show what the offspring will look like. You can also just kind of do this with some simple math, right? So we're going to talk about predicting the results of crosses, right? So kind of skipping the Punnett square and just doing the numbers. So it's going to take a little bit, just a little bit of probability, right? So probability is just statistically predicting outcome of random events. All right. So if things aren't random, it's much more complicated. If things are just happening randomly, right, due to chance, we can run some numbers basically. Now, we've determined, right, principle of segregation, principle of independent assortment, the behavior of the chromosomes in meiosis is independent, right? They're assorting randomly and they're assorting randomly independently of each other. So it's a series of random events, more or less, which means we can use probability to predict the outcomes of crosses. Probability values range from 0 to 1. You can kind of think about it like percents, right? You have a 0% chance of something happening or a 100%, right? So a chance of 1 means an event is certain. It's like a 100% chance. a probability of 0, basically a probability like 0, you have 0% chance that an event could ever happen. Right? And we can express these as fractions, which is kind of nice because it's going to look like the Mendelian ratios we were just looking at. So we can kind of line one up to the other. Okie dokie. Easy example is a coin toss, right? Coin has 2 sides. You have 2 possible outcomes. It's going to be heads or tails. So the probability of heads, you have 1 in 2 chance. 2 options. One of those things is going to happen. The probability of getting heads is 1 in 2, or 1/2. Right? That's not bad. You can just swap them out for genotypes. It's fine, right? Big P, little p, functionally just heads or tails, right? So probability of gamete with the dominant allele with a capital P is 1 in 2. It's just the same. All right, so cool. Some events are mutually exclusive, right? Which means they can't happen at the same time, right? You can't flip a coin and get heads and tails, right? It's not what happens. But you can flip a coin twice and get heads and tails, right? Because the 2 coin flips are independent events. Okay. Okay. So we can make kind of some rules about how to calculate probability, looking at independent events. Rule 1, rule of addition. For 2 mutually exclusive events, right? We're just talking about, right? Flipping heads and tails, inheriting a big P and a little p from the same parent. For 2 mutually exclusive events, the probability of either event happening is the sum of the individual probabilities. All right, so let's use dice. So we've got some more options. Dice. Dice have 6 sides. You've got 6 mutually exclusive outcomes. Right? You can't roll a die and get a 3 and a 4 at the same time. Well, yes, that's harder math. We're not doing harder math. We're just adding. Right? So any outcome, anytime you roll a die, is 1 in 6. All are equally probable. So in a single roll, I have 2 mutually exclusive events, rolling a 2 or a 6. But what is the chance that I roll a die and get a 2 or a 6, right? Like either option would be good for me. Not yet. So this is the rule of addition. So if rolling either a 2 or a 6, so the chance of rolling a 2, 1 in 6, chance of rolling a 6, 1 in 6, add them together, 2 of 6. You have a 1 in 3 chance when you roll a die of either getting a 2 or a 6. All right, so rule of addition. Well, we can put genotypes in there. So we've got this heterozygous purple F1 cross. I'm missing my X, but you can pretend it's there, right? So this heterozygote crossed with this heterozygote. There are 4 possible outcomes, right? The offspring can inherit both dominant alleles, dominant and recessive, recessive and dominant, or both recessive, right? Those are our 4 gamete options from this cross. There are 4 mutually exclusive outcomes. So the probability is 1 in 4 for any of them. They're equally likely. Yeah. So the probability that when I make this F1 cross, that my F2 offspring are a heterozygote of either option, right? Could be a heterozygote with this genotype or a heterozygote with this genotype. Each one got a 1 in 4 chance. 1 in 4 chance that it's big P, little p. 1 in 4 chance that it's little p, big P. 2 in 4 chance that you get either or of those. So there's a 1 in 2 chance that you have a heterozygous offspring. Right? So you have, right? So 1 in 2 or half. If you remember the Punnett square, right? We had homozygous dominant, purple flower, homozygous recessive, white flower, and 2 heterozygotes, right? In the other 2 squares. It was a 50% chance, 1 in 2. 2 of 4 were heterozygotes, right? So it matches the Punnett square, which it should. If it didn't match the Punnett square, we'd have a problem. All right, so think. Yeah, gets us to rule of multiplication, and we're right at time, so we'll pick that up on Monday. All right,
Nice.
sit with that a little bit if you have time.